Hello, welcome to MOOC video on vitrification. Term vitrification is derived from the Latin word vitrum meaning glass. Vitrification is defined as a transformation of a substance into glass. Vitrification is one of the popular techniques applied in cryopreservation of any biological material. Historically, vitrification of a biological substance has been performed many decades ago, starting from 1938 on frog sperms by Basil J. Lewitt, a professor of biology at St. Louis University. Lewitt showed that at rapid cooling rates, supercooled liquids can become so viscous that they can solidify without crystallization, forming a transparent, amorphous, glassy state. Interestingly, Lewitt became the founding president of Society for Cryobiology in 1964. Later in 1980s, Craig Fahey published the celebrated photo of a vitrified rabbit kidney. More recently, other organs such as rat liver, heart, and organisms such as Drosophila zebrafish have been shown to vitrify, as shown in photo. To understand vitrification, let's look at something called a phase diagram of cryoprotective agent CPA. Phase diagram is a plot of transition temperatures as function of concentration of a CPA. An example plot is shown on left. To vitrify a CPA, one can either follow equilibrium vitrification, where we follow the liquidus curve in phase diagram, or one can follow non-equilibrium vitrification, where one rapidly cools from a stable liquid state to below the glass transition temperature curve, as shown in blue arrow. In order to successfully vitrify, a rapid cooling is required to avoid any ice crystallization inside nucleation and growth region. Let's take a look at different scales of vitrification and cooling techniques used at each of those scales. On the table, you can see sizes starting from 1 microliter to all the way up to liter scale system. Okay, so let's dive into each one of the size scale. First, we have a cryotop, which can hold from 0.1 to 1 microliter volumes of sample. Secondly, we have a capillary straw corresponding to tens of microliter volume. Third, here we see a cryo mesh which can cool 100 microliters and larger sample volumes. Now, moving on to larger scale, a cryo valve with a capacity of roughly 1 milliliter can be used to vitrify samples. Or we can use 50 ml conical tubes for milliliters of sample volume such as rat organs. Lastly, as we scale up in a vitrification size, we have a cryo bag with a capacity of 0.5 liter corresponding to a clinical scale human kidney. Now, to successfully vitrify a biological sample, two major challenges needs to be overcome. First, ice formation, which can occur if cooling is not fast enough in ice growth and nucleation temperature range. Second, fractures, which can occur due to thermomechanical stress by non-uniform cooling. Now let's start from microliter vitrification. Cryotop is a highly conductive platform attached to the end of a rod, as shown in right. So here we can see a cryotop which can typically handle 1 to 4 microliter of sample volumes. Now first we will load a sample droplet on the cryotop platform using a pipette, after which the cryotop is plunged into Allen-2 bath, providing you a vitrified droplet. Now, for slightly larger sample size, a traditional straw can be used in the range of 10 to 100 microliter volume size. You can see a straw here. Straws are usually very thin for achieving faster heat transfer to the sample. Now, to load sample in a straw, it is dipped inside the sample, wherein the capillary action fills the liquid of sample inside the straw volume. Now, These straws are quickly quenched into cold liquid nitrogen bath. This, in turn, produces a vitrified glassy state of your required sample. Further larger samples in range of hundreds of microliter can be handled using a cryomesh, as shown in photo. Cryomesh is a mesh made up of highly conductive material with low thermal mass, allowing enhanced heat transfer to the sample using conduction, also eliminating any lead and frost effect. First, sample droplets anywhere from 100 microliter to larger are placed onto the mesh, after which cryomesh is quickly plunged inside liquid nitrogen bath, allowing a rapid cooling rate. Finally, as seen here, vitrified droplets of the sample appear clear and transparent, indicating a successful vitrification. Now, we move on to milliliter to liter scale vitrification. First, we'll see a cryovial with a 1 milliliter capacity. A cryovial can be directly plunged into LN2 bath for rapid cooling. Let's look at a conical tube which can contain up to 50 milliliters of volume. These volume can be relevant for cryopreservation of various biological systems such as tissues, blood vessels, small animal organs like rat kidney, rat liver, and rat heart, etc. Now, traditional approach of cooling where a conical tube containing sample plunged into LN2 bath would lead to fractures or physical cracking due to large temperature gradient across the sample size. Therefore, a controlled cooling rate would be needed, as in a control rate freezer, for successful vitrification with the conical tube. Lastly, for clinical scale human organs vitrification, we need multiliter volumes ranging from 0.1 to 1 liter. Here we show a cryobag which has two main advantages over traditional rigid tubes. One, cryobag is a clear container with flexible boundaries allowing free thermal expansion of a vitrified material. Second, its thin wall allows faster convective cooling to the sample inside. Here we will use a control rate freezer for cooling purposes. A control rate freezer, or CRF, is a cooling device where sample is cooled using a flow of liquid nitrogen vapors using forced convection mechanisms. This device can maintain a desired cooling rate by controlling the flow rate of LN2 vapors inside the cooling chamber. These LN2 vapors are supplied via a larger liquid nitrogen tank. So, for 0.5 liter sample volume, we choose a cryobag with a 3D printed 1:1 human kidney replica inside in it. After loading the sample, cryobag is hanged inside the CRF cooling chamber. A cooling protocol is then implemented, with first step being a rapid cooling from start temperature till glass transition temperature, which ensures no ice formation. After reaching glass transition temperature, an annealing step is executed, wherein the sample is held long enough at same temperature for thermal equilibration of hole geometry. Lastly, very slow cooling rate is chosen in glassy region below glass transition temperature till storage temperature to prevent any thermomechanical stress fractures. At last, let's summarize what we have learned in this video. Vitrification is glass formation which can be performed from microliter to liter size scale using different techniques from cryotop, straws, cryomesh to cryovials, cryobags in a control rate freezer. Ice formation and fractures are two major failure modes which need to be avoided for a successful vitrification. Some of the mechanisms involved in cooling techniques currently used for vitrification are boiling, free and forced gaseous convection, etc., as listed on the right. The plot on the left shows the maximum center cooling rate of a sample achievable by a certain cooling technique at a given size of biosystem. Note that as we go larger in size of a system, the cooling rate decreases, highlighting that either higher concentration of CPAs is needed for successful vitrification at these larger scales, or the unmet need of discovering a volumetric cooling approach. Here are some relevant references related to this video. Now, we have come to the end of our video. Hooray! Thank you for listening to this MOOC video. Hope you have a fun learning. Bye-bye.